I'm Akshay Anand. I am Associate Professor at the Department of Neurology at Postgraduate Institute of Medical Education and Research in Chandigarh, a city which is about 220 kilometers northwest of New Delhi. And it is one of the preeminent medical institutes in India, and I run this neuroscience research lab with a focus on neurodegenerative disorders. I'm also the editor-in-chief of Annals of Neurosciences, which is the Indian Academy of Neurosciences quarterly journal. It's a free online access journal. And I will take any questions you have on our new research on ALS, myolateral sclerosis. And hopefully, if there is any feedback, I will be happy to take more questions thereafter.
Great. So a series of studies suggests that people in India tend to show the first signs of ALS earlier, but live longer with the disease, nearly 10 years on average, compared to about 2 to 3 years elsewhere in the globe. What made you first suspect that the neuroprotective substance, VEGF, might be the secret, quote-unquote, to slower progression of disease in this population?
Yeah, the very fact that the Indian ALS patients live longer than the Western counterparts came from our clinical counterpart and chief of the department, Dr. S. Prabhakar, who pretty much sees the patients in the outpatient clinic, and also from the previous study published from the South India. In India, we have very few epidemiological studies. So from whatever data that comes from the published study in South India and from clinical colleagues, we found that the overall mean age of these patients, yes, 10 years plus, and which is not as what is found in the Western world. So this itself prompted us to look at the patients. We could not take their cords. The autopsies are very difficult to come by. The patients they find it very difficult to donate their bodies or cords. So we took the CSF, the cerebrospinal fluid, and the blood serum to see if one of the very key molecules in the angiogenesis and neuroprotection, which is vascular endothelial growth factor, VEGF, was playing any role in offering neuroprotection. And the origin of the study came from our own lab's ongoing neurovascular focus in determining various other ocular and neurodegenerative disorders, where we have been actually sorting out VEGF-R1 expressing stem cells and testing them in a variety of animal models of stroke, of retinal degeneration, memory loss, and so on. And this new role of VEGF, it is being reported from all around the world, in particular Peter Kamlet's lab in Belgium, prompted us to look at the role this particular molecule may be playing in enhancing this mean survival of the patients in India. So that's how we got started with the sampling of the serum and the CSF from these patients, presenting in various forms of clinical severity, and then tried and see if there is any association with the genotype, clinical phenotype association, if it is there or it is not there. So that's pretty much the reason why we wanted to look at the VEGF in these patients.
And you found a difference, right?
Yes. Yeah, we actually analyze the protein levels using ELISA. ELISA is a standardized technique to look at the protein expression of the secretion capacity of any biofluid you are investigating. In this case, it was CSF and serum. And we found that the VEGF was upregulated in the patients which were of clinical severity, which was higher than the ones which are just beginning to get ALS. And this increased apparently with the severity of the disease. And the VEGF levels were normalized to total protein, as is done in all the standardized, established ELISA assays. And we found there was significant upregulation of VEGF as the severity of the disease went up. And we hypothesized that this could be due to a compensatory effect, which is why we speculated and discussed in our discussion in that paper which came out in Journal on Neuroinflammation as to it might be offering, the EGF may be offering neuroprotection, possibly leading to an enhanced survival time in the ALS patients. And our comparison, our control group was, when we looked at the VEGF levels, it was the neurological controls, which means the patients having disease of the brain of degenerative kind, but not having ALS. And then we later on, the paper that came out later after this JNI paper was in European Journal of Neurology, when we thought we should, it was a logical follow-up of our own finding that VEGF was upregulated, to look at its scavenger receptor, which is SFLT or VEGFR1, which is very much known and was recently reported to have a direct role in keeping the cornea white, because it could keep the VEGF's pro-angiogenic action at check by scavenging action. So the VEGF and the VEGF receptor SFLT1, which is a free-floating receptor, keep each other's expression level in sort of a self-regulatory check, or they have they apparently share an inverse relationship. So more is the VEGF, it has been reported, less is SFLT1, and vice versa. And we did this in our patients, and when we compared them with the control group, we found SFLT levels were expectedly low, and this apparently seemed to us as a possible biomarker or a prognostic marker for ALS patients, which has not been reported before. So we are very excited about this data, and we are progressing further with analyzing at the RNA level of these biofluids from these patients versus controls, and looking at other things like PO2 level and POCO2 level to address queries if this could be due to hypoxia or not. So you need to show the PO2 and the POCO2 level in the arterial blood. Sorry, in the venous blood, yeah.
So you said a lot of things there. So stepping back just a little bit, you're thinking then that what's happening perhaps in these people with ALS is that the VEGFR receptor drops in level, which therefore boosts the level of VEGF over the course of the disease, and then in some sort of compensatory way helps to, keep those motor neurons alive, reducing the neurodegeneration in those patients. That's what you're thinking.
Yes, pretty much this, yes, because the VEGF is not only known to be neuroprotective, it's also very widely known from Judah Folkman's work at Harvard to be a pro-angiogenic molecule. So it results, or it helps in neovascularization and new blood vessel formation. And all neurons, for that matter, will be happy to have more supply of nutrients coming from new blood vessels. And it is this arborization of these vessels that feeds the motor neurons to keep healthy, and any disruption in this supply of blood through these new vessels resulting from VEGF's overexpression can lead to degeneration. Well, that is a hypothesis, but more studies need to be done at the molecular and the animal model level to show the real direct influence of SFLT to motor neuron degeneration.
Okay, so getting to that question, what else do you think really needs to be done to sort of show that this VEGF and VEGF receptor 1 could be the reason behind the slow progression of these people with ALS? What are you thinking of?
So the very first thing that needs to be done to take the study forward is to even before going to the molecular level of how SFLT is impacting the health of motor neurons, is to do a larger study within India as well as a cross-cultural study between low-time survivors and high-time survivors, between India and Caucasian population, for example, maybe a thousand patient study, a collaborative study between, let us say, US and Indian patients, and then segregate them into those who have a lower mean survival time and a higher mean survival time, and then compare the SFLT levels and the VEGF levels, in the sera and the CSF of these two populations. And then going from the leads that are established, because we have to realize this is a small study. Even though the ALS incidence is low, it is significant from the statistical standpoint. But a larger study is obviously desirable. And once that study comes up and the results are in our hands, I would do something that we are actually already simultaneously embarked upon, is to collect CSF from these patients from across the country and store them and use the CSF as an inducer to an in vitro growing motor neuron cell cultures. And these are NSC34. These are the famous motor neuron cell line. And this has been used by scientists around the world to look at the behavior and the in vitro characteristics of these motor neuron cells, motor neurons in culture. And the idea is to look at if CSF is having SFLT expressed more or less, or VEGF being less or more, should impact the NSC34 cell line, motor neuron cell line, in the manner, in a way that it may either lead to enhanced apoptosis or reduced apoptosis. So the first question would be to look at a range of patients, a group of patients having, let us say, a highly upregulated VEGF level, and how this impacts the health or the survival time of the motor neurons growing in culture, versus another group of patients that are having a low VEGF, and looking at how the cells behave in terms of surviving in dish, in culture, versus SFLT with the different groups of groups of patients having varying range of varying concentration of SFLTs, and then test how these NSC34 cell line that we want to employ, and in fact we are writing a grant application to do just this, and investigate and examine if what is the impact on the viability of NSC34. This is a first way of looking at the impact of such molecules coming from CSF from the patient directly. So that's something I can think of immediately. And there are, of course, other related plans which I, for obvious reasons, because it's a grant application, I cannot divulge. But we'll be happy to say something which can impact patients and the community of ALS.
Right, of course. So you're thinking then a combination of both larger epidemiological studies to sort of confirm that this is really a difference between a population like in India, slow progressors, right, versus where there's what we see in the States, for example, where you see more of a range of people. And at the same time, do the correlative biochemical and cell biological experiments to also look at that mechanism, look at that correlation of neuroprotection, see whether or not that it's really sort of correlating between, what you're thinking about over the course of disease by comparing people with definite ALS versus probable ALS versus possible ALS versus controls. That's what you're saying, I think.
Yeah, that's pretty much the content of the next grant application. And let me add that we also want to look at the emerging biomarkers in ALS, as it is happening around the world for any disease. You look it up in the PubMed, the search engine for all the scientific publications, and you come up with new biomarkers. And the ones which are coming up in a big way, you might, I'm sure, be familiar with. TDP-43 and optineurin is coming up in a big way, at least in the way in terms of publications that are coming up and showing up in PubMed. So we are in any way trying to, since we're going to take the CSF and the blood from these patients anyway, so we will be investigating the levels of these other emerging markers as well, and see if they are in any way converging towards a common anti-apoptotic pathway in which VEGF is known to play a significant role. And we are also combining, and this is a huge effort actually, trying to put the autopsy studies as part of our live patient study, which is very, very difficult. In India, it's really very difficult to get the patients to agree to donate their bodies or spinal cords for us to investigate and see if what we are seeing in CSF and in serum is actually showing up in the autopsies, which is the crux of the matter, the real evidence. So we are collaborating now with Daisy Sahani, a new head of the Department of Anatomy at the same institute. She's a very dynamic lady, and who's doing a lot of social work, going out to people, influencing them, and talking to them, and telling them what good comes to the community if they donate their bodies, for example. And that's an effort we are doing very as a in a very top priority level.
Right. So again, you're thinking looking bigger, more people, getting out there in the community, really trying to ask the question of whether or not this does correlate and sort of ends at late stage end of disease to sort of
Yes.
clinch this hypothesis. And also, as you're saying, looking more globally, thinking about ways that you can leverage these findings to the people with ALS at large by looking into the idea of this being a biomarker for prognosing the disease, for monitoring patients and getting a sense of what their clinical diagnosis might look like. The idea being that, given how much VEGF receptor 1 you might have, you may be able to tell a lot about what's going to happen to that patient, but at the same time inform clinical trials for evaluating clinical therapies.
That's right. And that's why I think through this medium, we can reach out to ALS scientists and volunteers in US and around the world. Whoever looks at your ALS Development Institute's profile, and whoever are your people connected to your network, we look forward to collaborators in that community to join hands with us and together do this multicentric study, as I would like to call it, and investigate and find out, who knows, this could be the real molecule suppression or reduction or inhibition would actually lead, if not to curb the fatality of the disease, in at least extending the survival time.